Well, hello again, everybody. Welcome back again. We are now into the home stretch as far as the semester is concerned, because we are beginning our look at the last kingdom that we need to look at this semester, and that's the animal kingdom. We have 4 chapters devoted to that kingdom. One is a fairly easy chapter to comprehend, and that will be the next chapter that we'll be looking at, chapter 32, which is a brief introduction to the animal kingdom. Then we have the longest chapters and most energy-dense chapters that we will deal with this semester, and that's chapters 33 and 34, which cover the animal kingdom, the invertebrates first in chapter 33, and then the vertebrates in chapter 34. But before we begin that more detailed look at the animal kingdom, we start with a chapter that looks at embryological development with respect to animals. And you might be asking yourself, why do we choose to do this chapter? Why is it included in the semester? And the reason is fairly easily summarized by a truncated term, which I'll explain to you in a moment. And that truncated term is Evo-Devo. Sounds like a really bizarre, maybe some strange 70s alternative rock band. In fact, it's a relatively new discipline within biology that has emerged over the course of about the last 15 or 20 years, I guess. That is the merging, I guess is the best word, of 2 existing disciplines, evolutionary biology and developmental biology, to create this strange beast that we refer to as evolutionary developmental biology. And the reason why is because if we want to look at the evolutionary origin, particularly of animals, then by looking at the development of the embryos and looking at how similar embryological development is and where embryological development diverges from the common pathway and where along the common pathway that divergence occurs, gives us some clues as to the evolutionary relationship of different groups of animals. And a lot of this is being driven by DNA sequence analysis. We've already talked about the fact that the evolutionary history of any organism is written in its DNA, and it has been looking at that and looking at the role of genes in embryological development and animal development in general that has led to a renewed interest in embryological development as a window on evolutionary origins and evolutionary relationships. And so in animals, very particularly, this is something that you can look at relatively easily, because there are ways of generating animal embryos in vitro, in glass petri dishes and the like. And in the last 20 years, that technology has even become applicable to looking at human development with the rise of in vitro fertilization techniques, because there, the early embryological development does take place in petri dishes until you have an embryo at a stage that is implantable into either the mother or the surrogate mother's uterus to complete the developmental process. And so that's allowed us to take a look at vertebrate and particularly mammalian animal development in lines of animals where previously we wouldn't have even had a hope of looking at that early embryological development. So this has become a very, very powerful tool. And it is one of the most definite cutting edge areas of biology at the moment. And we'll dig a little bit into some of the cutting edge part of this, but I'm going to try and avoid getting too deep into it, because if I do, I'm probably going to number one, create a very long recording. These things get long enough as it is, as you already realize, but it's also going to take us away from some of the more fundamental things that you really need to be aware of out of this chapter. So I will delve a little bit into some of the details where I think it's appropriate for you to know a little bit more than the textbook has provided. And there are some sections that in this new edition of the textbook that we have just adopted that have been left out that I think are worthwhile including. So you're going to find some material that's in here that you're probably not going to find in the textbook. So when we get to that, I'll let you know, and you might want to pay a little more attention to that because you're not going to find a detailed explanation in the textbook. So with that out of the way, let's get started on looking at animal development. So one of the fundamental questions that people have asked once we were able to actually start taking an interest in our own origins as individuals was just exactly how does the single-celled zygote, that product of the merging of an egg and a sperm, develop into a very complicated multicellular organism with some of those complexities already starting to emerge within the first trimester of pregnancy. In fact, by week 12 of embryological development, all the fundamental body systems are in place. And really the last 2 trimesters of human pregnancy are devoted to maturation and enlargement. So everything is at the end of week 12 is very, very primitive, nonetheless recognizable for what it will eventually turn into at the end of fetal development just prior to birth, but these are in very primitive forms. And so the last 2 trimesters are all about maturation of the organ systems and enlargement of the organ systems, with the lungs being the last system to mature, which is the reason why premature babies have so many issues, because gas exchange via the lungs is the last system that needs to be developed in order for the embryo or the fetus, if you want to refer to it that way, in its more well-developed form. It's the last system that needs to develop before the fetus is capable of completely independent existence, independent of the mother. And so when there is a risk of premature birth, one of the things that the OBGYNs and the prenatal specialists focus on is trying to force the maturation of the lungs through steroid injections to the mother. And if there's still no hope of preventing premature birth, once the baby is put into an incubator, again, it's placed on a protocol that supports the poorly developed lungs and tries to help increase the rate of maturation and therefore give the premature baby a chance of survival. And we've got pretty good at that. But it still begs the question, how do we get to that stage from this single-celled structure, the zygote? And that's really what we're going to talk about in this chapter, or at least we're going to talk about the 4 recognizable steps that we identify that constitute the early steps of embryological development. So we're not going to go all the way from conception to birth, but we are going to go probably until along the embryological developmental timeline, if we're thinking in terms of humans at least, until around week 8 to about week 10, when we can confidently say that organogenesis is well and truly underway. That is the development of organ systems. That begins a lot earlier in human embryological development, but we do recognize that by about week 6 to week 8, you can clearly start to recognize the major body systems, the organ systems, because they're starting to develop at this point. That means the last step of embryological development, organogenesis, is well underway. So we are going to start with fertilization, the first step, when egg meets sperm and what takes place there. Then we'll talk about the 2 intervening steps, cleavage, and a very, very important 3rd stage called gastrulation. Of the 4 stages, gastrulation is probably the most significant. and we'll spend some time talking about the significance of gastrulation. And once we've worked our way through all of these 4 steps, then we're going to focus on some steps that are not necessarily included as part of these 4 embryological development stages, but they are really important because they really talk about why you wind up being an organism containing approximately 10 trillion cells and about 220 different types of cells, all derived from that single zygote. So there's some important ideas here to be considered as we work our way through this chapter. So very early on in thinking about how this process might have taken place, before there was any really concrete scientific study, there were 2 prevailing ideas that people proposed as an explanation of how a human being, a fully developed human being in the form of the baby at birth, could develop from what was clearly individual cells, the egg and the sperm, which were well known. And one idea is this idea of preformation, which basically says that inside the reproductive cells, either an egg or a sperm, depending on who you talk to, there was a tiny little perfectly formed miniature person, which is referred to as a homunculus. And if you take a look at some of the diagrams that were published in some of the early books on embryological development coming from the 18th and early 19th centuries, these are the sort of pictures that you would see. Now, it is somewhat easy to understand why people would draw this conclusion. If you have nothing better than a very primitive light microscope to look at sperm cells, the best you're going to see is a rather amorphous looking interior of the sperm cell, and the same thing is true of an egg cell. So basically you have the freedom to speculate pretty much whatever you want on what could be contained within a sperm cell or an egg cell that contributes to human reproduction. Excuse me. And so this was one of those ideas, that you have a completely formed miniature human being encapsulated in one of these cells. Now, it doesn't take much to realize why this is clearly not true, apart from the fact that we have much better microscopes. We can look inside the cells and we can see quite clearly that a homunculus is not found in the interior of human reproductive cells. But logic dictates that this is probably an idea that should never have been proposed. Because if you have a completely formed miniature human being inside a sperm cell, the implication of that is that inside this completely formed, mature, but miniature human being must be reproductive cells that in turn contain a tiny, mature but miniature human being. And basically you go back, and the question you have to ask is a sort of philosophical question: do fleas have fleas? And do those fleas have fleas ad infinitum? It's an exercise in straining human logic and human imagination. But this was nonetheless a very popular idea. The competing idea, which is sort of diametrically opposed to this idea of preformation, is an idea that was first proposed by Aristotle. And the reason why it has traction, or had traction, in Western thought was because of Aristotle's contribution to Western thought during the Reformation, when Aristotle and Plato and the Greek philosophies were rediscovered by the introduction via Muslim thought of Aristotle's thoughts into the Reformation movement. So the Greek philosophies were never lost during the quote unquote dark ages. They were well preserved in the Muslim world. And part of the Reformation, or probably what kickstarted part of the Reformation, was simply the rediscovery of all of these works that have been preserved in the Muslim world that represented the Greek thought that had influenced the Romans and subsequently influenced Muslim thought, and then ultimately influenced Western European thought via the Reformation. And Aristotle said that basically you start with a blank sheet. That the, if we want to use the proper term, the zygote represents a completely formless precursor to whatever the organism is going to become. And through a series of progressively more complicated steps, ultimately this blank slate, this formless egg or formless zygote, would develop into the baby at birth. And because of Aristotle's influence over Western thought, this was also a very prominent idea. So you have these two competing ideas, what you might refer to as diametrically opposed. One says that all the information for forming a human being is completely enclosed within the reproductive cells, pre-formation. The other one says, no, the reproductive cells contain no pre-existing information, and you start with a blank slate and you progressively develop a fully developed human being in a series of stepwise processes or stepwise developmental stages. It turns out that the whole notion of pre-formation probably has some merit, even though in terms of what Aristotle might have said about a blank slate and looking at an egg cell and a sperm cell seems to suggest that that might be the situation. Pre-formation may actually have some merit because we've discovered that, in fact, there are molecules within the egg cell that are placed there by the mother that really do represent information that will determine the development of the subsequent zygote / embryo. And so the egg really does contain some pre-existing information. And we refer to these molecules, and they could be in the form of proteins, messenger RNAs, a combination of both, as determinants. And these determinants are inserted into the eggs in a significant number of species. And we'll look at some examples along the way as we work our way through the presentation today. And so as much as pre-formation in the formal sense, the homunculus, we now recognize makes no sense at all and is not a good explanation of embryological development, we can't dismiss the idea that some pre-existing information is passed on, particularly via the mother, through these determinants within the egg cell that in the end do play a role in determining embryological development and the fate of the cells that are produced during embryological development. And as I said, we'll get into a little bit more detail about this as we progress through the presentation. So once we recognize that we do have to come up with an idea of how embryological development can take place, going from a single cell to a multicellular organism, in the case of most animals, then we also need to recognize that there are two fundamental processes that are going to take place. One is called cell differentiation, and we'll also talk about a partner process called cell determination. We'll get into those probably about 2/3 of the way through the presentation. But both of these are important because if all the zygote does is grow and divide and produces identical daughter cells, then all of those cells would be exactly the same. And we'd see no cell specialization, we'd see no tissue development, no organ systems, because all of the cells would be not only genetically identical, which we know is the case for all of the cells in our body, but they would also be functionally identical because there would be no specialization. So at some point, cells do need to take on specialized roles, which is referred to as differentiation. they must also have some sort of knowledge about where they lie within the three-dimensional structure that is the organism. And that three-dimensional positioning information will determine what type of structures they'll become part of. And then there needs to be some way for them to determine what specialized cells they will become within that structure. And this is the notion of determination and differentiation. Along the way, we also need to be able to explain morphogenesis, the process by which the animal's body takes on a very, very specific shape. So are there reasons why, for instance, most animals are bilaterally symmetrical, have a head at the anterior end of the body, and have limbs that are, if we're talking about terrestrial tetrapods, attached to the axial part of the body. So our arms and legs are attached to the axial part of our body, which extends from the anterior, our head, to our posterior, our butt. And so we have an axial structure and an appendicular structure, the arms and legs being the appendicular structure. And when we look around and take a look at other animals, we see that they have either similar shapes if they are terrestrial tetrapods and related to terrestrial tetrapods, or they have other body plans on the basis of what is appropriate for them. But we need to be able to explain how these body plans develop. This is the process of morphogenesis. And so we'll be focusing on this as well, this process as well, and the accompanying processes that fit in or that give rise to morphogenesis. Let's state it that way for now. So a lot of the information that we're going to talk about in this chapter is not gained by direct observation of human embryological development. As I said, we've only really been able to do that for about the last 20 years or so. In vitro fertilization actually has about a 40-year history approximately. But it's only in the last 20 years that we've been able to utilize embryos that are not wanted anymore for in vitro fertilization because the couples have got the children that they want, and any excess embryos under normal circumstances would likely have been either destroyed or, and this sounds pretty, yeah, but this doesn't sound nice, but a lot of them actually wound up being flushed down the drain because they were no longer necessary. No one really had an idea of what to do with them. But people interested in embryological development, and particularly human embryological development, realized that these unwanted embryos that were being kept in liquid nitrogen against the day that they might be needed, once it was determined that they were no longer needed, represented a source of embryos that could be used to study these early steps in human embryological development. So we have access to that information now, but much of what has been determined big picture about embryological development has been gained by using organisms where it's very easy to watch the early stages of embryological development because of the way in which it takes place, primarily because fertilization is external, it takes place in water, the eggs are large, or they're accessible in the case of the chick. And so a lot of the information that we're going to talk about from this chapter is actually from these model organisms. Remarkably, however, much of what we have learned from these model organisms we now know translates pretty much almost identically with some modification to mammalian development and specifically to human development. Where there are significant differences, we'll stop and talk about those significant differences along the way. So let's keep going. So as I mentioned, there are 4 stages of development: fertilization, which most people are aware of, when egg meets sperm, and we'll talk about what happens when that takes place. It's not quite as simple egg meets sperm, zygote forms, on we go. It's a little bit more complicated than that. But once the zygote is formed, then that cell and the resulting cells undergo a process called cleavage, which results in ultimately first a solid ball of cells referred to as a morula, and this is usually in the human situation considered to be around 16 cells to 32 cells. At that point, this solid ball of cells starts to expand, and a hollow space opens up in the center of it, which is referred to as a blastocele, and the resulting structure is referred to as a blastula. The cells that are formed during this process are called blastomeres. So you can see that there's a new vocabulary to be mastered here. And again, as we saw with the plant kingdom, there is a vocabulary of words that sound very similar. And that's because they relate to similar structures. So cleavage results in the blastula. And we'll talk about the nature of cleavage, because it's not the normal type of cell division, because that's what cleavage refers to here. It's not the normal form of cell division that you might be familiar with from Biology 1, where you learned about mitosis. It's a little bit different. The next step, or step number 3 of the 4, is gastrulation. And as I hinted at earlier, this is probably the most significant of the 4 stages, because here you reorganize the embryo and you generate 3 layers of tissue known as germ layers. And it's from these 3 layers of tissue that all the organ systems are going to emerge. And different organ systems are associated with each of these germ layers. And it's really important that you come to terms and know which organ systems are associated with which germ layers. And as you will see in this chapter, there is a table, excuse me, it's color-coded because there has been an effort to coordinate the color coding of this table amongst the textbook publishers and also the supply companies that provide the models that people use to illustrate these processes, so that there is a common coloring scheme. So when we get there, you'll see that the color patterns are very distinctive, associated with each of the tissues or the germ layer tissues. And it is really important that you pay attention to which tissues and organ systems are derived from each of the germ layers when we get to that point. Then the final stage is organogenesis. This is where the 3 germ layers give rise to the different organ systems. And we'll focus on some of the early organ systems to develop and look at how reorganization of tissue layers and reorganization of cell layers leads to the development of some of the early organ systems. So here you can see the 4 stages of embryological development. And then once we get the beginning of organogenesis, then we move on into larvae and adults in the case of the frog, one of our model organisms, or we could say moves from embryo to fetus to baby and adult ultimately. Depends on what type of organism you're talking about as to how you would label these stages. But these are the steps that we are going to focus on in this chapter. So let's take our first break, and I will stop here for a moment. And I'll see you on the other side of the break with the answers to these questions. So what 2 apparently incompatible ideas have historically been used to explain embryological development? And question 2, list and briefly describe the 4 stages of embryological development. I'll see you on the other side of the break.